Hello everyone, welcome to the channel Microbial World. Today we are going to study about scanning electron microscope SEM. So the scanning electron microscope, it is an improved model of electron microscope. It is used to study the three-dimensional image of the specimen. And as we have said before, the scanning electron microscope, it uses electrons instead of light to form an image. Okay, so these electrons, they interact with the atoms that make up the sample and they produce signals that is used to form the image. So now let's look into the construction of scanning electron microscope. So the scanning electron microscope, it consists of an electron gun. As we have said before, the electron gun, it consists of a cathode and anode and it produces high energy electron beam. Then there is a magnetic condensing lens. Okay, so the magnetic condensing lens, it condenses the electron beam. Then there is a scanning coil that is arranged between the magnetic condensing lens and the sample. And the function of the scanning coil, it is to deflect the electron beam over the specimen. Then there is an electron detector scintillator. So this electron detector, it collects secondary electrons. Okay, so these secondary electrons, these are the electrons that is emitted from the specimen. Okay, so they collect secondary electrons and convert it into electrical signals. Then there is a video amplifier and cathode ray tube. So you can understand these parts more clearly when we explain the working principle of SEM. Now let's see the working principle of scanning electron microscope. Okay, so you can see here, this is the electron gun, grid, magnetic condensing lens, scanning coil, sample, collector, scintillator, video amplifier, cathode ray tube, scan generator. Okay, so the electron gun, it will produce a stream of electrons. Okay, so these electrons, it is called primary electrons. And these primary electrons, it is accelerated by the grid and anode. And the accelerated primary electrons, it will pass through the magnetic condensing lens and the scanning coil and will be incident on the sample. So when an electron beam, it hits the sample, some of the electrons, it will be backscattered. Okay, so these are backscattered primary electrons. Then there will be X-rays, Auger electrons, secondary electrons will all be emitted from the sample. So these secondary electrons, okay, so you can see here, these are the secondary electrons. Secondary electrons, it will be collected by the detector, okay, or the collector. And these secondary electrons that is entering the detector, it will strike the scintillator and will emit light splashes. Okay. It will strike the scintillator and emit light splashes, and a photomultiplier tube, it will convert these light splashes into electrical signals. Okay. These electrical signals, it is amplified by video amplifier and then it is fed to the cathode ray tube that will produce an image that can be viewed and photographed. Okay. Advantages and disadvantages of scanning electron microscope. So the advantage is that it is easy to operate and they also have a user-friendly interface. They also have variety of industrial applications, okay, that is to analyze the surface of solid objects. And some of the modern scanning electron microscopes, they can generate digital data that can be portable. And also it is very easy to acquire data from the scanning electron microscope. Okay, we can acquire the data within a short period of time of about 5 minutes and all. And the disadvantage is that it is expensive to purchase and they are also bulky to carry. They also must be used in rooms that is free of vibrations and free of electromagnetic elements. And they must be maintained with consistent voltage. They also should be maintained with access to cooling systems. Applications of scanning electron microscope. So SEM is used to study the filament structures of microorganisms. It is also used to study the topography of elements used in industries. Then it is used in the analysis of cosmetic components which are very tiny in size. It is also used for spot chemical analysis in energy dispersive X-ray spectroscopy. These are the applications of SEM. So this is all about scanning electron microscope. Thank you.